Hi, my name is Angela Robinson, and I'm going to take you on a tour of the animal cell. The animal cell is a eukaryotic cell. A eukaryotic cell has membrane-bound organelles, linear chromosomes, and a nucleus. They are much larger than prokaryotic cells, which lack these characteristics. These small bound organelles allow compartmentalization for various functions to be separated, but their work is interdependent. Let's begin our tour. This is the endoplasmic reticulum. It is composed of sacs and tubules which modify lipids and proteins. The ER is composed of two compartments. The smooth ER are the small yellow figures on the pink structure that helps synthesize lipids, carbohydrates, and detoxify substances like alcohol. The rough ER is the light pink membrane between the tubule sacs. It helps synthesize new proteins after translation by adding side chains. All eukaryotic cells have a nucleus. This is where DNA is housed. In the nucleus lies the nucleolus, where RNA is transcribed like mRNAs and rRNAs. Inside the nucleus, DNA is safely housed in a complex. The complex is in an unwound pack called chromatin or heterochromatin. It can't be transcribed. However, during transcription, the complex histones unravel euchromatin, which can be transcribed. When we zoom into the nucleus, we see small holes called nuclear pores. When modified mRNA leave the nucleus, they pass through the double membrane nuclear membrane envelope. The mitochondria is the final area where most of the ATP from oxidized glucose is catabolized when oxygen is present. It is a double membrane structure which increases surface area for electrons to power ATP synthase to generate ATP. The mitochondrion is considered endosymbiotic. Earlier, I said that it was a double membrane structure. The outer membrane of the mitochondria has eukaryotic proteins, while the inner membrane, which makes the cristae for the matrix, is composed of prokaryotic proteins. Circular-shaped DNA, which are found in prokaryotes, are also found in the matrix. The yellow figures are called lysosomes. They have low pH enzymes that can break down many macromolecules like proteins and lipids. The small yellow spherical organelles are called ribosomes. They synthesize proteins during translation when mRNA from the nucleus are read through the ribosome to code proteins. For example, in the pancreas we have a lot of ribosomes, where insulin, a hormone, is secreted to lower blood glucose levels. The green spherical structures are called peroxisomes. They have enzymes that break fatty acids and amino acids. These enzymes safely break down peroxide into oxygen and water, which accounts for any oxygen radicals that could damage cells due to instability of electrons. The yellow rod organelles are called centrosomes. They are found only in animal cells. They pull replicated chromosomes in opposite directions to divide the cell. Near the ER is the Golgi apparatus. Proteins, lipids, and carbohydrates that have gone through the ER may be sent to the Golgi for further processing. When we go back to slide 5, secretory and membrane proteins are modified in the rough ER. These compounds enter through the cis and exit through the trans in or on vesicles, where the Golgi attaches carbohydrates to the modified protein. The vesicles are transported to the cell membrane to form either peripheral or embedded proteins. Cells are mobile, and cytoskeleton gives cells their structure. Some cells have a flagellum or cilia which help project the cell in different directions. Cilia are tiny projections, while flagella are long tails. The cell is confined in a selectively permeable membrane called the plasma membrane. It is comprised of phospholipid bilayer, which allows certain molecules to pass. Its layer is made out of phospholipids, which is comprised of two fatty acid tails, which are hydrophobic, and a phosphate head that is hydrophilic. The model that characterizes the cell membrane is called the fluid mosaic model. What makes the plasma membrane mosaic are the carbohydrates, cholesterols, and membrane proteins. The embedded and peripheral proteins make up part of the membrane proteins on the cell membrane. These proteins can serve as enzymes or identification markers in the cytoskeleton system, which prevents the immune system from distinguishing them as invaders. Some proteins are embedded in the membrane to serve as passages to facilitate the movement of molecules using channels and carrier proteins. Channels like aquaporins help move polar water molecules across the hydrophilic regions of the protein so that it enters the cell. Carrier proteins allow molecules to bind to the protein membrane and induce a conformational change, allowing the molecule to enter the cell. Different charges of ions outside and within the cell establish a concentration gradient, which requires energy to move ions across the cell membrane. This is called active transport. Cells have protein pumps that help move ions against the concentration gradient. In animal cells, the sodium and potassium pump creates an electrochemical gradient when potassium enters the cell and sodium leaves the cell. The pump is a protein which goes through conformational changes when ATP releases a phosphate which binds to the pump. In the first stage, 3-sodium binds to the pump from the inside of the cell. A phosphate changes the pump's orientation and the 3-sodium flow out. Next, 2-potassium binds to the pump from the extracellular fluid. The phosphate is released from the pump, which reorients and causes potassium to enter the cell. Well, that's the whole tour of the eukaryotic animal cell. As you can see, eukaryotic cells are very complex organisms. My name is Angela Robinson, and I hope you have enjoyed our video.